Okay, everyone. I hope you guys had everything go well with the quiz. And as usual, if you have any questions, please let me know. Today's lecture, this is labeled as lecture 6 and 7, and it's because there's going to be some practice involved. So we're going to go through the lecture, and then I'm going to ask you guys to work through some practice problems, because the best way to learn this is to actually do it, okay? So today we're focusing on systematics. There is an entire field of science devoted to this, okay? And basically what this is it focuses on the diversity of life, past and present, and then the relationships among the living things through time. Okay, so you tend to visualize these on a tree. They call them an evolutionary tree or phylogenetic tree, or any number of those. They have two components. They show the branching order, which kind of shows who's closest related to who, and then they show branch length, which shows the amount of time or the amount of evolution that's actually gone by. So within systematics, there's a whole bunch of different branches or subsets, okay? And so taxonomy is one of them. And so taxonomy is basically the practice of classification. And you can think of taxonomies or taxonomic schemes. They're basically made up of taxonomic units known as taxa. We tend to use taxa singular or plural, even though technically taxon is singular. And basically what we're doing is we're arranging things in a hierarchical structure, okay? And so the whole idea of kingdom, phylum, class, order, family, genus, species, nature does what it does, but we tend to classify it in that way because it's the way we understand it the best, okay? So, like one species doesn't care how we've designated it or designated a second species, it does its thing, but basically, this is how we can keep track of things, just as for our brains. So I do want to know a little bit about the history of systematics and the way people used to think, because again, we take this knowledge for granted, but the reality is things have changed drastically over time, okay? So back in Aristotle's day, now remember Aristotle, he was arguably the founder of biology, and what he felt was that the adult would represent the final attainment of the embryo. That was the teleos, or the ultimate. Although sometimes these days, being an adult, I kind of think I'd rather go back to being a kid. That's just me. Anyway, but that was Aristotle's chain of thought back in the day. So back in Aristotle's day, the way they used to think and the way they sort of used to organize things is known as the ladder of nature, or the great chain of being. Okay, and so the way this works is that each form represents a link or a rung in the progression from the most imperfect, which is the most primitive, to the most perfect. Okay, and so if you go look at the upper right corner, that's the great chain of being. So you have non-being. Okay, then you have minerals and plants. Poor plants, they always get the short end of the stick. Okay, bad joke, I know. Then you get to animals, and then you get to eventually you get to angels, and then you get to God, and that's the absolute most perfect form. So again, that was just how they used to think of things to try and keep it in order. Now, Carolus Linnaeus was an interesting guy, and he definitely was the father of modern systematics. And so he came up with a method that we've been using now, for quite a long time. So he died in 1778, came up with the whole idea of kingdom, phylum, class, order, family, genus, species, and he was famous for classifying critters. Okay, and so he definitely had an interesting personality. From what I heard, or what I was told long ago, was that he had a bit of an ego, okay? And so, again, interesting guy. And so when it came time for him to publish his work, he actually put a picture of himself on the front cover of his book holding an apple in the Garden of Eden. Now, in this day and age, you would get yourself laughed out of the city, of science, if you ever even considered doing that. And so we, tend to not do those things. But again, this was back in the 1700s, and well, that was his thing. So when he published this work, he basically outlined what we know about species and animals and plants, and kind of developed the Linnaean hierarchy of taxonomic levels. So remember the kingdom, phylum, all of that came from Linnaeus. And, we appreciate it, and we still use it for the most part today. But things have definitely changed just a little bit. So, of course, the way this works is kingdom, phylum, class, order, family, genus, species. Now, I don't know what you all used to remember this, but when I was trying to learn it, King Philip came over from Greece Saturday. Okay, so and of course we're going to have open note, open book tests, so it's not like you have to memorize this, but you might want to end up knowing it for the future. Okay, and so what you see here is an example of humans and how we happen to be classified using Linnaeus' system. Research has shown, by the way, when you can use little tricks like King Philip came over from Greece Saturday, it really helps you learn things. And I can tell you I've remembered this for, decades, we'll say. So, I definitely agree with that. Now, back decades ago, when they were teaching about the different kingdoms, we had the plant kingdom, the fungal kingdom, the animals, the protista, and the monera. Okay, and so, again, this was built upon his system. Basically, he came up with plants and animals and then wasn't really sure what to do with everybody else, kind of, sort of, but he kind of knew that there would be more kingdoms that would develop over time. So along came another scientist by the name of Carl Woese, who would basically take a gene that every single living critter had, and he would determine the sequence of all of those critters, as many as he could possibly sequence. And then he sort of took all of the data, compiled it, and tried to piece together a tree of life. Okay, and you got to give him credit. This was back in the 70s when sequencing took forever, and you had to have a lot of patience. And so he went through and did this and actually won him several prizes. I'm probably going to put up a video for you guys to see. One thing I'll tell you I don't like about the video is the fact it kind of makes him sound like he's full of himself. I was actually super lucky when I was in school and I got to meet him because he actually taught at the University of Illinois when I was a grad student there. And he was one of the nicest people I'd ever met. And I was a student. He didn't care. He was super nice to me no matter what. And so he might come off as a little pompous in the video, but that wasn't the man that I knew. One more quick interesting story about him. He'd actually sort of achieved the fame that I never will, and most of us never will. He won so many awards for coming up with the whole idea of domains, by the way. So the domain eukaryotes, bacteria, and archaea, these domains were based on his data. And basically the way this works is it sits above kingdoms, okay? And it's kind of wonky, and it's not perfect, but it works. And according to the scientific data, this is how things sort of evolved. But anyway, so he had won so many awards for coming up with this particular paper that when it came time to teach, he basically could just teach whatever he wanted to, and the school let him just do it. I'll never get to do that. Okay, I'll never achieve such fame. But again, he was a nice guy, so I was super happy for him. Now, Charles Darwin, as you all know, came up with the idea of natural selection. And so basically what he did was he would propose some of the first trees, and he had a drawing of it in his book Origin of Species, okay? And so remember, he was kind of challenging the conventional thought at the time, and so he knew that, but he was a really good scientist, and he had a pretty good idea of what was going on. Now, I'm sorry about the weird formatting. Definitely make sure you download your PowerPoint slides because it's got the correct versions. But two things I want you to know the difference between. I want you to be able to define them and compare them, okay? So analogous trait is a trait that is superficially similar between a couple different groups, but they derived independently through convergent evolution. And if you recall, we kind of talked about that in the very first lecture of class. So insect wings and bat wings evolved completely differently, totally different physiology, totally different mechanisms. However, having wings is a really useful thing, and so it happened multiple times in evolutionary history. That's analogous. Now, homologous, remember homo means one, okay? And so a homologous trait is similar between two different taxa because a common ancestor had it and then passed it down. Okay, so the digits in bat wings, by the way, is the same as the digits that we have in our fingers. And once again, as my husband's trying to shush the dogs behind us, I'm reminded why I miss my office. Now, there are not 3, 4 general, I really need to fix that one of these days, general mathematical models for how you classify your critters, okay? And I'm not expecting you to, don't memorize this. Just know there's 4 different ways, and we're going to zero in on one in particular for this class, but realize that the way you put your trees together differs, and there's a couple different ways to do it. One of them is called phonetics, where you look at the similarities between the taxa and you group your taxa accordingly, okay? Another method is called cladistics, and this is the one that we're going to look at in depth. Now, that is when you put your groups together based on what are known as shared derived traits, otherwise known as synapomorphies. You need to write that down. Even though it's listed here, I want you to write on your notes, synapomorphy equals shared derived traits, and then put it in your own words, okay? Because we're going to see that term a lot for the rest of this lecture. Another way to put these trees together is to use what's called maximum likelihood. And basically what this does is it incorporates, so the computer knows that mutations happen a certain way. Some are more common than others. And so it kind of puts those probabilities in those models when it's calculating the tree. Okay, so certain mutations are more likely to happen than others. It takes that into consideration as it's looking to see what taxa are related to who. All right. Last but not least, if any of you are die-hard mathematicians, you might have heard of Bayesian probability. And so basically, Bayesian probability, the best way I can think of to explain this is you're taking a priori knowledge, or knowledge that you know ahead of time about your trees, and you're incorporating that knowledge into this algorithm or this equation as you put together this tree. So that's why it's called a posteriori probability. So you're taking these probabilities of what you already know and you're incorporating that into your tree. Okay, and I realize it's kind of complex. Again, you don't have to know this in depth, okay? Just realize that trees can be put together many different ways. Now, the one method we're going to zero in on is called cladistics, okay? And this was invented by a man named Willy Hennig. He died in 1976. I was alive then, but I don't remember any of it. So that's all we're going to say about that. Now, cladistics is based on a couple different grouping patterns, okay? So, and I want you guys to know these, and what's going to happen is the next slide, you're going to pause lecture and you're going to circle an example of each of these, okay? So keep this in mind as we're going through, so make sure you understand this. So, monophyletic group contains a common ancestor and everybody that derived from that ancestor, okay? Mono means one, so there's one common ancestor and then everybody that came from that. Clade, that's basically part of a phylogeny that's monophyletic. This is the clade, then if you say clade, everybody knows that is monophyletic. Just another term for it. Polyphyletic. Now basically, a poly means many. Okay, so a polyphyletic group contains creatures that did not necessarily all evolve from the same common ancestor. Now, for those of you that are super detail-oriented and you tend to think about things in depth, I realize you could come up with the argument that all of life came from one critter, okay? But the reason we have these terms in this field is so systematics people can talk to one another, and as soon as you see the term monophyletic, you know what that is. You can't study everything, okay? So you can study, though, a polyphyletic group, and as soon as you see polyphyletic, you know that means that you have a couple of different common ancestors with critters that came off of them, okay? Last but not least is paraphyletic. And so paraphyletic means you have a common ancestor and some of the taxa that came from that common ancestor, but not necessarily everybody. So just think of it that way. Okay, so now what I want you all to do is pause lecture, and I want you to come up with an example on this tree, and I want you to circle it, of a monophyletic group, a polyphyletic group, and a paraphyletic group. Okay? So pause lecture, circle these, label them, and then start up when you're ready. Now, an example of a monophyletic group, otherwise known as a clade, could be the red group, or the green group, or the yellow group, okay? It's just a common ancestor and everybody that came from that common ancestor, all right? Now, a polyphyletic group would mean multiple common ancestors. So we might think of the red group and the purple group, okay? Or the red group and the yellow group. There's multiple common ancestors. And I realize if you go all the way back, they all have one common ancestor, but don't overthink this. Keep it simple. It's just so people can talk to one another, okay? Another example then of a paraphyletic group, okay, would be, let's say in the red group, it's the Arctic fox, the kit fox, the red fox, the fennec fox, raccoon dog, and bat-eared fox, but you leave out a few. Okay, so that's paraphyletic. So you have some of the same group, but you don't include everybody. Make sure that that makes sense, because later on we're going to revisit this, and you got to be able to know what each of these is. Now again, these trees are put together based on what are called synapomorphies, and these are shared derived traits, which means the common ancestor has them, and that everybody that came from the common ancestor had it. Okay, so it's a shared derived trait. That's what a synapomorphy is. Now, the thing to remember is that cladograms are rooted, and so basically you include what's known as an outgroup, okay? So the whole idea is the outgroup is that crazy uncle that no one wants to claim, okay? Or that crazy cousin that no one wants to claim in their family. And so you know that it's related to this group, but it's more distantly related than who you're classifying. And the whole purpose of using an outgroup is that it kind of gives your traits polarity. Because you know if the outgroup has it, but then a couple of your members inside the group don't have it, then you know there might be a newly evolved trait that those other critters have and the outgroup doesn't, if that makes sense. So again, you're just trying to find polarity. So what's derived, what's new versus what's ancestral. Whatever the outgroup has, that's ancestral. Anything that's new relative to the outgroup, that's going to be a derived trait. Okay, so I'm going to have you guys, I'm actually going to include this link in the announcements, so all you have to do is click on it, and I want you to read through this because they do a really nice job to describe how you put together a tree. And the reason I'm harping on this, by the way, is because, hint, hint, the next assessment you guys have, i.e. test, hint, I'm going to ask you to put together a tree. Okay, and so the other thing you guys are going to do today, lecture is not super long, and you only have one of them. Because this usually is the lecture for 2 days, is I want you guys to practice putting together a tree. And I want you to feel super comfortable with it. Okay, so the reason is because later on in the course, we're going to see tree after tree after tree. And I want you guys to have this down cold, that you know exactly how to read it, you know exactly how to put one together, and all sorts of other good stuff, that you're really comfortable with it. Now, I can tell you, I remember a while back, and it was a long while back, when people started to use genetics to put trees together. And initially, for the longest time, people only used morphology, okay? But there's pluses and minuses to using morphology, and we'll talk about that in the next lecture. When people were using genetics, they thought, oh, finally we're going to solve all the questions and we'll know the tree of life and be able to piece together everything of who's related to who. Well, just like every other field of science, this had its share of problems too. And it definitely was not the end-all be-all when it came to putting your trees together. So honestly, the best method is to use morphology and genetics, realizing that sometimes they disagree. And when that happens, you have to find another way to address those questions. Now, we've said before that morphology is definitely not perfect, especially when you're trying to put together your tree. One reason is that very often it's hard to tell what is ancestral and what's derived. So what's the polarity? Secondly, it's really hard to tell if certain traits are analogous or homologous. Okay? And last but not least, some characters are morphologically correlated. Okay? And that's going to really mess up your data if you're trying to treat them as independent when you're putting your tree together. One really good example I can think of is heart development and limb development are actually really correlated. I have a very close friend that went into limb development for research, and apparently in mice, when there's heart defects, there's limb defects, a lot of the time anyway. So when you're treating them as independent, though, that's going to mess up your tree if in fact they're correlated. Now, one of the ways you can actually address these questions is to study developmental pattern. So in other words, the evolutionary development. And there's an entire field of science focusing on evolutionary development and how different traits evolve and have developed and so forth. And so the things you have to remember is that complex characters are likely to have a complex developmental pattern. So it's highly unlikely that that exact same pattern is going to develop more than once. Okay? This is why, you know, a lot of aerobic critters are assumed to be related, because the aerobic mechanism in physiology is so complicated. The likelihood of having that evolve multiple times is really unlikely. Okay. A second way that this is interesting is that developmental patterns will very often reveal correlations between morphological traits. So how one limb develops, you know, can then reveal things about how the heart develops, for example. So when you study how a creature is developing and you look at the patterns, that can address these things. Okay. Additionally, last but not least, if you can figure out the development, that very often will help you clarify the polarity, what's ancestral and what's derived. All right. So I don't know if you guys ever took an embryology course before. I took one in college, and that gave me such a headache, because scientists have names for every little speck and every little dots on the slides, and goodness, it gave me headaches. But it was interesting. Okay, just gave me a lot of gray hair at the time. Now, DNA isn't perfect either, and we've said this before, and so it has its own host of problems when you're putting together your phylogenetic tree. You only have 4 character states, okay? So A, G, C, and T. And so you can have a mutation and go from A to G, G to C, C to T, and T goes back to A, and you'll never detect it, all right? Something else that might cause problems, different genes evolve at different rates. That was something that scientists had just sort of figured out when I was in grad school, and they were totally shocked. They thought that all genes evolved at the same rate. Well, it doesn't happen that way at all. Okay, so if you have a gene that evolves really quickly and you get certain results, they're going to be different results than if you have a gene that evolves really slowly. Okay, hybridization can really mess up your trees because you don't know if your differences are due to the fact it's a different species or if it's a mutation within that species, which that's going to change your tree. So only fixed differences are informative, and when we mean fixed, we mean everybody in that species has it. Okay, so if you're looking at a trait and it's not fixed within a species, it might be the same as something else, and then it just confuses everything. Okay, so some lineages will actually mutate quicker than others. This is where we say molecular clock ticks unevenly into the different lineages. I can tell you spruce trees mutate really, really, really slowly. That was what I used to do my research on, and finding mutations in spruce trees was really hard because they live for 200 years. Okay, however, fruit flies mutate really quickly, as evidenced by those flying around in my kitchen. Just kidding, sort of. Anyway, those guys mutate quick because their generation time is really quick. Okay. Also, some patterns of DNA evolution can cause certain algorithms to have the wrong answer. Okay, so in other words, transition, transversions, the type of mutation you have may not agree with the model you're using. If you have recombination, okay, genetic recombination in nuclear genes, that can mess things up. And then for number 8, cross it out. We don't need to worry about that one because I ditched the figure to go with that one. So basically what it, don't worry about it. Okay, so now what I'm going to do for the rest of our time, because I realize this lecture is, we have some time left, is I want you guys to practice with this, okay? So I am going to put up some videos that I want you to watch because I think they're good and they will show you how to go through and put together your tree. And then I'm going to have you guys practice and put a tree together, and if you turn it in, I will give you an extra credit point. Okay, so I will put a place for this. I'll make sure it's up there by tomorrow sometime, Friday sometime. And if you guys have any questions, let me know. I wish you a wonderful, safe, and healthy weekend, and I will talk to you again on Monday.